origin of life, of the tremendous turmoil in this area, that is among the evolutionists. I think the creation scientists probably should be given some credit for that, because we certainly have challenged these people. And your organization here and others, all just like this, all over the country have done this. And the amazing thing about it is this. You know why Darwin won most of the scientific world to evolution? It certainly wasn't because he presented any real good solid evidence for evolution. He couldn't, the fossil record didn't support his theory. He knew that. He said that one of the greatest evidence against his theory was the fossil record itself. The missing links, you know, they call them, well, they're still missing today. We haven't found those transitional forms that his theory required. You know what won most of the scientists' point of view was the conviction that Darwin had provided a mechanism. He finally had described the means by which evolution could take place. It seemed so logical and so plausible and workable that it won the scientific world, most of them, over to his point of view. And for 130 years, hundreds of thousands of scientists have worked on that theory and on that mechanism. The incredible thing today is that many evolutionists are abandoning Darwinism. They're giving up on Darwin and his mechanism. They finally realized that it didn't work and couldn't work. They finally realized what the creation scientists have been saying about that theory for 130 years is true. Now, I'm not saying that all scientists, all evolutionists have abandoned Darwinism. No, not at all. There's a lot of them going to cling to it. But more and more scientists are saying that Darwin was wrong. And yet, you know what? They still say evolution is a fact, evolution is a fact, evolution is a fact. We all know that evolution is a fact. It's just the mechanism that's in question. I'd like to know how they know it's a fact. They can't produce the historical record, the fossil record to support it. They don't have a mechanism. It reminds me of what Al said to Pogo one day. He said, you know, we could have bacon and eggs for breakfast if we had some bacon and if we had some eggs. And that's the way these evolutionists have a good theory. They had some fossils and they had a mechanism, but they don't have either. Here's an article that appeared in Newsweek, that wonderful Science Weekly. It's called, this was April 8, 1985, more than 3 years ago. The article is by Sharon Begley, called Science Contra Darwin. In other words, science against Darwin. In her opening sentence, she says, the great body of work deriving from Charles Darwin's revolutionary 1859 book on the origin of species is under increasing attack, and not just from creationists. Then she describes some of this turmoil and arguments that's going back and forth, tearing at each other's throat, so to speak, among evolutionists. And then she says this, so heated is the debate that one Darwinian says there are times when he thinks about going into a field with more intellectual honesty, the used car business. My apologies to any used car salesman in the audience. I want to remind you, it was not a creationist who said that, it was an evolutionist who said that. I have a book here by Dr. Michael Denton. Dr. Denton has 2 earned doctorates. He has a PhD in molecular biology and an MD from British universities. He's now in genetic engineering research in Australia. He's establishing himself as a world-class expert in genetics. This man is not a Christian. He's not a professing creationist. But his book is a devastating critique of evolution theory. On every count, according to Dr. Denton, evolution strikes out. The title of the book, Evolution, A Theory in Crisis. Let me read just a few comments on the flap of the book. It says here, the theory of evolution as propounded by Darwin and elaborated into accepted fact, quote unquote, by biologists, is in serious trouble. This sober, authoritative, and responsible book by a practicing scientist presents an accurate account of the rapidly accumulating evidence which threatens to destroy almost every cherished tenet of Darwinian evolution. Later, we read, not only has paleontology failed spectacularly to come up with the fossil missing links, which Darwin anticipated, but hypothetical reconstructions of major evolutionary developments, such as that linking birds to reptiles, are beginning to look more and more like science fiction fantasies than serious conjectures. And then finally, we read, most important of all, the discoveries of molecular biologists, of whom Michael Denton is one, far from strengthening Darwinian claims, are throwing more and more doubt upon the correctness of the whole theory. You know, what's so valuable about this book is that Denton, not being a Christian, he's a fine gentleman. I've last 2 visits to Australia had a chance to spend some time with Denton. Fine gentleman. He's not a Christian, and he makes no profession of being a creationist. He doesn't even mention the creationists. That is the post-Darwinian creationists. But he's saying the same things that we creation scientists have been saying for years. And they can't accuse him. You know what they do to us? They cannot refute the scientific case that we present. So they try to destroy our integrity. They say that we quote out of context, that we distort science and outright lie and so forth and so on. All these charges they make against us to try to destroy us, because they cannot change the laws of science and they can't produce non-existent transitional forms, so they can't refute our evidence. Well, they can't make that charges against this man Denton, you see. He's not a Christian, not even a professing creationist. Yet he says the same things that we're saying. And I think he says it even better because he's such an excellent writer. Now finally, let me call your attention to the fact that a well-known Swedish scientist has published a book last year with the title, Darwinism, the Refutation of a Myth. Darwinism, the Refutation of a Myth. This man is Søren Lovestrup, very well-known British embryologist, Swedish embryologist and scientist. I met him in Jerusalem several years ago. He and I were invited to Jerusalem as speakers at the first annual conference on origins and evolution. And when I heard him speak, I realized he's not the ordinary evolutionist, because he rejected Darwinism, this idea of the evolution through mutations and natural selection, just totally rejected it. And he said the evolutionary changes have occurred very abruptly, suddenly. He embraced Richard Goldschmidt's hopeful monster mechanism, which in essence, Goldschmidt and Otto Schinewolf, very famous German paleontologists who also suggested this, that the first bird hatched from a reptilian egg. And that's why we have no transitional forms between a reptile and a bird. There just weren't any. Of course, they found it very difficult to convince people that something like this really did happen. But this was the suggestion of this man. So he is against Darwinism. He says the mechanism won't work. It didn't work. And so he's desperate. I think this is desperation to suggest something like this. But here it was one of his closing statements in this book. Now remember, he's a very well-known Swedish scientist. He is an evolutionist. He is not a creationist. And this is what he says on page 422 of this book. Quote, I believe that one day the Darwinian myth will be ranked the greatest deceit in the history of science. End of quote. Let me quote that again. I believe that one day the Darwinian myth will be ranked the greatest deceit in the history of science. Just think. This Darwinian myth, Darwinism, is what is being taught in practically every major university and college in the United States, and in our schools, and in all the popular media, National Geographic, and things like that. Embraced by thousands and thousands of scientists and supported by the vast majority of scientists for 130 years. And if this famous Swedish scientist is right, and we certainly think he is, it's the greatest deceit in the history of science. Now we go beyond that, of course. We think not only is Darwinism such, it is wrong, it is totally wrong, it has deceived millions and millions of people, but the theory of evolution itself is the greatest deceit in the history of science. If God created us, as we are certain he did, God should receive the glory and the honor as our master and our creator. How could evolution be anything but a deceit or a lie of Satan? I certainly would not accuse individual evolutionists of being dishonest or being the tools of Satan. I think they're just dead wrong. But Satan is using the theory, you see, to deprive God of his honor and glory as our creator. Well, let's take a look now at this specific aspect of origins. You know, many times I meet evolutionists in my debates and so forth who say that we shouldn't include the origin of life in evolution. That's not part of evolution. The only thing in evolution is the origin of species. You start with something living and then you have to explain how all of us got here. But you'll find chapters on the origin of life in practically every biology book. And you find in most of these books theories on the origin of the universe and so forth. Now, these people don't like to talk about the origin of life. They say, no, it's not part of evolution. You know why? They know how bad off they are, how much deep trouble they are in their theories on the origin of life. They don't even want to talk about it. They just say, it's not evolution, let's talk about something else. But it is part of the evolutionary continuum, you see. If you want to talk about, say, that first cell evolving into man, well, where did the first cell come from? You got to account for that too, you see. Then you got to account for the earth, where it evolved, and then you got to account for the universe, you see. Now, many years ago, I read a book by a man named John Kiyosin. And he was very optimistic at that time, this is 1964, about the future developments in this area of the origin of life. He felt that we were on the right track, that scientists would work this out. It wouldn't be long before we'd know the answers to some of these critical aspects of the problem of the origin of life. Well, he published an article in 1978, that's 14 years later. Now we find that he was anything but optimistic. He was totally pessimistic. This article was published in Origin of Life, 1978, beginning on page 569. Now this is what Kiyosin says. The claims of chemical evolution are unreal. We are asked to believe that biochemical compounds, biochemical reactions and mechanisms, energy metabolism and storage, specific polymerizations, codes, transcription and translation apparatus, and more appeared in probiotic waters, that is, before life existed. That's what he means by probiotic waters. Appeared in probiotic waters with the function they would have in a living thing before there were living things. Of course, he said this is unreal. They wouldn't have those functions, you see. Later he says, all present approaches to a solution of the problem of the origin of life are either irrelevant or lead into a blind alley. Therein lies the crisis. Really, you know, now that comes from a man who's an origin of life chemist, a man who worked on the problem. And it's true. All of these things that these people do are exercises in organic chemistry. That's really all. They're irrelevant as far as the origin of life are concerned. And they lead into a blind alley anyhow. Now maybe we have that first slide, please. And we'll need to dim out. The first slide is not very good. I apologize for this first slide. The rest will be a lot better than this. Now, if you're going to consider, anybody's going to consider this origin of life question, you have to go through several stages. We'll get a few more lights back on here in a minute. But for this first slide, we'll turn them off. You have to, the origin of life chemists have to think about this problem in stages. Now, first of all, he has to imagine what the primitive Earth, what his hypothetical primitive Earth was like. What kind of gases were in the atmosphere? what sort of energy and how much energy was available? What was the temperature in the hypothetical primordial ocean? What kind of chemicals were dissolved and what was the pH and so forth? Now, it's strictly hypothetical. Obviously, there were no human witnesses there. And so he tries to infer what the earth might have been like. Now, you can bet he's going to do one thing. He's going to imagine things were favorable for the origin of life, naturally. That's one thing he's going to include. Well, that'd be the first step. Now, the next step would be trying to imagine the origin of rather simple chemicals, like amino acids and purines and pyrimidines and sugars. They're really not simple, but compared to protein and DNA and RNA, they are simple. How, under these assumed primitive earth conditions, would these compounds be generated? Now remember, we have 350 million cubic miles of water on the earth. Everything eventually is going to be dissolved in that water, diluted. You can imagine how a pond might dry up and concentrate chemicals or something like that. But that's only temporarily. Eventually it rains and washes that stuff right out in the ocean. So you see, whatever generates this stuff has to be extremely effective and efficient. It has to generate billions of tons of each individual molecule that contributed to the origin of life. Not the total quantity of material, but each individual molecule must be generated in billions of tons. And so we must try to imagine some rather efficient methods, you see. Not generating a few parts per billion. They talk about seeing molecules out in space. Well, they do that by the absorption of light, and I'm really rather suspicious about all this. But anyhow, if there are molecules out there, a few parts per billion or trillion, well, we're talking about billions and billions and billions of tons of these things you see here on Earth. Now, the next slide, please. Oh, I have the control here. All right. Now, the next thing we have to try to imagine, now we've got the ocean full of amino acids and things like that. Then we have to imagine how complex chemicals were generated, like ATP and DNA and RNA and protein molecules. Again, we've got to have billions of tons of each one of these individual molecules. Now, after we have generated somehow billions of tons of these very large and complicated molecules, we've got to imagine how they managed to get together and form these cycles, metabolic cycles that are part of a living organism. Now you see, that'd be difficult to imagine how that might have happened. We'll go into a little more detail on that, but you see, they have to get organized into these complex systems. Then the next slide, we see, they imagine then the first cell came into existence. Now, it's a tremendous leap from one of those macromolecules to the first cell. It's a leap of fantastic dimensions. And that's one of their biggest problems. How did we get that first living organism? Then on this slide, we see that they imagine the next thing, they have to solve the problem of reproduction. It doesn't work that way at all. Couldn't work that way. Reproduction had to be right there. The moment you had that first cell, it had to be able to reproduce itself. That's one of the criteria of life. A living organism must reproduce itself. No molecule does that. DNA does not do that. RNA does not do that. Viruses do not do that. Viruses are not living organisms. Viruses are large, complex RNA and DNA molecules surrounded with a protein coat. They produce no energy. They cannot generate the building blocks. They have no enzymes, nothing like that. They are not live. They have no metabolism. If you put viruses in a bucket with all the nutrients in the world, you'd never get another virus. A virus gets in one of our cells, like a polio virus gets in one of our cell reproduces copies of the virus. The mechanism of the cell is absolutely necessary for that. And of course, since it's making this virus, which is totally of no use to the cell dies. It's using its energy and material to make something that's totally useless for the cell, you see. So there's no self-replicating molecule. The only thing that replicates itself is a living organism. And reproduction requires the most complicated apparatus there could be. But it had to be right there at the very beginning, you see. Now, in the next slide then, they imagined that respiration then developed later. They imagined that life originated, as we'll see, in a world with no oxygen. And so oxygen was generated later as green plants developed and other organisms developed that had photosynthetic processes and could produce oxygen, and then oxygen accumulated in our atmosphere. Now, let's consider the type of atmosphere that now exists and must have existed if life evolved. Now, in the present atmosphere, we recognize the fact that there's about 98% nitrogen and 21% oxygen. That's 99% of everything. The other 1% is made up of mainly argon, with some water vapor, of course, and carbon dioxide, carbon monoxide, and very, very small amounts of other gases. Now, life could never have evolved in such an atmosphere. It's absolutely thermodynamically impossible. Why? If there were any sugars or amino acids or anything like that, these building blocks, you see, if there's anything like that around, oxygen would oxidize them and convert them into carbon dioxide and water and nitrogen and other compounds. You cannot have those coexisting, you see. You cannot have them in a living cell. Yes, the living cell has tight compartments, regulates and controls everything, no problem. But here on the Earth, just on the hypothetical primordial Earth, if you had oxygen present, it would in essence burn up everything. I don't mean with a flame. It would simply oxidize everything, just like iron. You know, iron spontaneously rusts. It's converted into ferric oxide. That's what would happen. So the origin of life chemist says, well, obviously there was no oxygen on the hypothetical primordial Earth. Instead of an oxidized or oxidizing atmosphere, called an oxidizing atmosphere, of course, because of the presence of oxygen, we had a reducing atmosphere, so-called because it had hydrogen rather than oxygen, free hydrogen. And in addition to carbon dioxide, it had some carbon monoxide and methane. In addition to nitrogen, it had ammonia and, of course, water vapor. And so they assumed then that the Earth had this sort of atmosphere. And as we will see, Stanley Miller and others performed all of those laboratory experiments on the origin of life using gases like this. But evidence has steadily accumulated in the past 30 or 35 years to show that the Earth never had such an atmosphere. It never had an atmosphere with methane, ammonia in it. Methane would be rapidly decomposed by ultraviolet light into nitrogen and hydrogen. If there's significant quantities of these hydrocarbons, there would be deposits of them in some ancient sedimentary rocks, not a trace. Now, furthermore, there's accumulating evidence. Not only did the Earth never have methane and ammonia in its atmosphere, but it has always had oxygen, significant quantities of oxygen. For example, a couple of Canadian geologists said, well, now the ratio or the distribution of certain minerals, the distribution of iron, the minerals of iron, carbon, sulfur, and uranium are determined by the amount of oxygen in the atmosphere. So they said, let's look at very young rocks, let's look at very old rocks. That is, very young sedimentary rocks, very old sedimentary rocks. If the concentration of oxygen in the atmosphere was different in the past, there'd be difference in the distribution of these minerals. And so they did. They looked at rocks which they thought were very young. They looked at rocks which they believed to be somewhere between 2 and 3 billion years old. Now, I don't believe in those billions of years at all, but they They thought they had sedimentary rocks that old or older, and they found there was absolutely no difference whatsoever. They said, therefore, from the time the oldest sedimentary rocks we can find on the earth, the atmosphere had just as much oxygen then as it has today. And there's much other evidence. Large deposits of sulfates, oxidized form of sulfur, banded iron deposits, which are, of course, the oxidized form of iron, and other things like that establish the fact there's always been a lot of oxygen in the atmosphere. So that settles it, ladies and gentlemen. Evolution could not have occurred. Life could not have evolved on this planet. Thank you. The lecture's over with. For all practical purposes, you see that establishing. But let's persevere nevertheless. Let's assume, shall we, for the sake of the argument, that the atmosphere of the, say, in the past was quite different than it was today. And let's see if life could have evolved under those conditions. Now, we have to account for such things as these, the purines, which make up the DNA and part of the DNA and RNA molecule, the pyrimidines, cytosine and thymine, which are found in DNA. You see, these are really not simple compounds, but we call them relatively simple. We had to have billions of tons of each one of these things, and the purines and pyrimidines, something's gone wrong with my pointer here. There we are. The purines and the pyrimidines must combine with phosphoric acid and with sugar to form a nucleotide. The nucleotide is the unit, the building block of DNA and RNA. So the building blocks of DNA and RNA are made up of three things, three types of chemicals: the purines and pyrimidines, phosphoric acid, and sugar. They combine to form the nucleotide. So we had to have billions and billions of tons of each one of these things. Now we also had to have amino acids. The typographical error here, and this slide I obtained from a biological supply house. They have oxygen here in place of carbon, but just imagine that's carbon as it is down here. These represent amino acids. They combine. We have two of them combined in the dipeptide, and then this amino group can combine with another carboxyl group, and this carboxyl group can combine with another amino group, and that can go on and on and on. And we have long chains of amino acids to make the proteins. So we must, of course, have billions and billions of tons of each one of these amino acids. Now, how did this happen? How on this primitive Earth, so-called hypothetical primitive Earth, was this accomplished? Well, they did a lot of talking, but nobody did any action until Stanley Miller performed his famous experiment in 1953. Now, Miller, assuming that the primitive Earth had these gases, methane, ammonia, water vapor, and hydrogen, put these gases in these apparatus. He had a boiling pot of water here that provided the water vapor. And here was an electrical discharge chamber simulating lightning. That was the energy source. Now you see, you have to have energy for the origin of life. Every step in the origin of life is an uphill step. You know that cars do not spontaneously run uphill. Now you can drive a car uphill, providing that you provide the energy, the gasoline. Now of course you and I recognize you got to have much more than gasoline, don't you? Got to have much more than just energy. You got to have a motor. If you pour a can of gasoline on a car and light a match, it doesn't go uphill, does it? It goes up in smoke. If you wanted to go uphill, you got to put the gas into a gas tank, which fed into a motor. The motor converts the gasoline or chemical energy into mechanical energy, you see. Well, Miller, he had to provide energy too. Now, so he provided this electrical discharge chamber, simulating lightning. Now, most of the energy available on the hypothetical primordial Earth, by the way, would have been the energy from the sun. Almost all the energy available would have been that energy from the sun. There would have been some energy available from lightning and very, very small amounts, relatively speaking, from heat, from radiometric decay and so forth and so on. But he used this apparatus simulating lightning. Now the gas is circulated through the apparatus, and every time a spark formed, it generated some of the products, which you noticed he had a condenser here, which condensed the water vapor, causing the water to condense and run down and collect into a trap, carrying with it the non-volatile products, while the volatile gases continue to circulate. After a week or two, Dr. Miller analyzed the solution here in the trap, and lo and behold, it contained a few amino acids, the building blocks of proteins. And in effect, the whole scientific world went wild. They said, look, here under plausible primitive Earth conditions, Miller has generated amino acids, the building blocks of proteins. I remember seeing an article in the San Francisco Chronicle about this. Headlines, scientists are near to creating life in a test tube. Why, that's the most ridiculous statement they could have made about it. Because I want to point out, first of all, the trivial nature of these results. What was produced? A few amino acids. Or in any similar experiment, you can generate a few amino acids or a few simple things. Now that's all that was accomplished. He was light years short of creating life, of course. You have to combine those amino acids to make proteins. You have to have DNA. You have to have RNA. You have to have complex carbohydrates. You have to have many other compounds. You have to have membranes. You have to have genetic material. You have to have energy factors. You have to have protein factors. And it all has to be put together in just a precise fashion. All he did is generate a few amino acids here. Very trivial results. And even those results would not have been accomplished if Dr. Miller had not imposed something on this apparatus that would never have been possible on the primitive Earth. That, of course, was the trap. You see, without that trap, Dr. Miller would never have obtained a detectable quantity of products. Why? Because those products, which he isolated in the trap, are destroyed by that electrical discharge at rates that vastly exceed the rate of formation. If the products had been allowed to come in contact with that energy, once again, the rates of destruction of those products would be more than 1,000,000 to 1. More than 1,000,000 to 1. And you would never obtain a significant or detectable quantity of product. But Miller provided the trap. Well, you might say, maybe there was a trap like that on there of some kind. Well, evolution has been thinking about that for years and they've never been able to come up with a trap yet. The ocean itself is not a trap. Now, they imagine some scenario like this. I took this out of a book on evolution. And you notice they have the gases here and then the electrical discharge or lightning and the ultraviolet light and form certain things which drift down to the ocean. The half time of fallout is about 3 years. That means if you have a pound of this stuff in the atmosphere, it takes about 3 years for 1/2 pound to reach the ocean. And then another 3 years for the half of the half to reach the ocean. In the meantime, while that's going on, it's being zapped by the lightning, burned to a crisp by ultraviolet light, and practically nothing reaches the surface of the earth. Glycine is the simplest amino acid. It's the easiest to form and the hardest to destroy. It has been estimated that 97% of that glycine would be destroyed before it reached the ocean. Now, even after those substances reached the ocean, they could not survive because ultraviolet light penetrates down through several meters of the water and is always continually stirred up. Furthermore, there are hydrolysis and many other chemical reactions taking place here which destroy those products. Now, if you take into account then these two processes, the rate of formation and the rates of destruction, and then calculate the net quantity of product formed, it is essentially zero, or very near to being zero. Very, very little product would be formed. It was calculated that glycine, its concentration in the ocean would be about 10 to the minus 20th molar. That is essentially infinite dilution. It would have no effect in any origin of life experiment. Glucose, you see, glycine only has 2 carbons. Glucose is a simple sugar, has 6 carbons. It has been calculated under these conditions. The concentration of glucose would be 10 to the minus 134th molar. That means if the entire universe was one big chemical reaction, you probably couldn't find one single molecule in the entire universe. Now, the principle here is very simple, but there's no way of getting around it. You see, you don't have to know any chemistry, you don't have to know any thermodynamics, all they have to know is this. The rates of destruction of these substances vastly exceed the rate of formation by any kind of energy that you would have on the hypothetical primordial Earth. And that's why right here at this stage, right here at the stage of the simple chemicals, you can destroy any notion on the origin of life. Ultraviolet light. Lightning is tremendously destructive. Look, you know, Miller, he has this simulating lightning, and he's supposed to generate life or substances leading to life. What happened to you when you get hit by lightning? Do you become more complex, more ordered? I'll say you get obliterated, don't you? You get burned to a crisp. Now that's what happens to all these substances. They're no different. If you want to kill bacteria, you put them in ultraviolet light. Kills them. Or viruses, or inactivate viruses. Very rapidly and very readily. Now here's another thing. If you could imagine a trap, supposing somebody could finally imagine a trap where you could remove the product, tramp out the product and save the product. Listen, that is fatal to any theory on the order of life. It's absolutely fatal. Why would it be fatal? What's the purpose of the trap? Remove the product from the energy. You're dead in the water. You can't go any farther. You can't do anything else. You don't have any energy. What happens if you're going down the highway in your car and you run out of gas? Can you go any farther? You just sit there, don't you? You can't go anywhere without energy, and the same thing is true here. How are you going to combine those amino acids to form protein when that requires a lot of energy? See how silly, irrelevant this sort of stuff is? And yet they teach it to our high school kids. Everywhere I go, even the question and answer periods, well, what about this experiment by Stanley Miller? And they're taught this stuff. And they're not presented these facts, you see. With the trap, they're dead, and without the trap, they're dead. I mean, they're just dead. That's all there is to it. Dr. D.E. Hull published an article in 1960, 7 years after Miller published these results. It's 1960. Happens to be 28 years ago. This is what he had to say in that article, published in Nature. Those of you who might be interested in reference, in Nature, volume 186, page 693. That's 1960. Now, this physical chemist said, quote, the physical chemist, guided by his fanatic religious ideas, that isn't what he said at all, is it? What Dr. Hull said was this: the physical chemist, guided by the proved principles of chemical thermodynamics and kinetics, cannot offer any encouragement to the biochemist who needs an ocean full of organic compounds to form even lifeless coacervates. End of quote. Now, a lifeless coacervate is just a blob of oily material that will separate out if you get a high concentration of these substances. They have no structure, no complexity, nothing like that. To get that, you've got to have an ocean full of this stuff. And based upon the proved principles of science, you can exclude that possibility, you see. Right here at this very start, we can exclude the possibility, and there's other difficulties. Amino acids and sugars combine with one another, and they destroy one another. The amino group of the amino acid combines with the carbonyl group, or the ketone group of the aldehyde, or the ketone or aldehyde group of the sugar. They combine. And that's the end of the amino acid, and that's the end of the sugar. Now, you see, a chemist will go in the lab and he generates amino acids by some method. Hallelujah, that's the way amino acids were generated. And some other chemist, he goes into the laboratory and sets up different conditions. He generates sugars. Hallelujah, that's the way sugars were generated. See, didn't happen that way on the primitive earth, did it? All happened together, all dissolved in this primitive ocean. They would destroy one another. Furthermore, there's an awful lot of calcium on the earth. Much, much more calcium than there is phosphorus. Calcium forms an insoluble salt with phosphoric acid. Calcite. Calcium, or calcium phosphate, is insoluble in water. It forms a rock that goes down to the bottom of the ocean and just sit there. All the phosphoric acid would be sitting down in the bottom of the ocean in the form of a rock, you see. All kinds of difficulties. All kinds of ways to make this, we know that this is impossible. But let's just assume, for the sake of the argument, we don't want to make this lecture too short, that we got an ocean full of amino acids and things like that. Well, here's a short section of a DNA molecule. And now here's the nucleotide again. Here's a pyrimidine, sugar, phosphoric acid. That's a nucleotide. There's a purine, sugar, phosphoric acid. You see, those are the nucleotides. Now, most DNA molecules have thousands of these nucleotides. At Berkeley, when I was with the virus laboratory, we were working with tobacco mosaic virus. I was particularly working with the protein part of it. But this tobacco mosaic virus, supposed to be a rather simple plant virus, has 6,600 nucleotides. One of the genes that codes for one of our blood clotting factors has 186,000 nucleotides. And if one of those is wrong, you cannot produce that blood clotting factor and you're hemophiliac. Now, I'm not saying that life had to start with a DNA molecule of 186,000 nucleotides. How in the world did we get that, you see, by chance? Well, in any case, we had to have the DNA molecule and the RNA molecules composed of thousands of these nucleotides. Now, we can see immediately one of the problems is you've got to combine these things in a chemical bond. The purine or pyrimidine combines with the sugar combines with phosphoric acid. That requires a lot of energy. That doesn't happen naturally and spontaneously. Those things just don't say, hey fellas, let's get hooked up here, let's join up. That doesn't happen at all. They don't want to do that. Those bonds break easily because when they break, energy is released. You know, if I'd asked several of you people to come here and join hands, you would have to consciously apply energy to raise your arm and clasp hands. It would require an input of energy. That's the way it is here. You must supply a lot of energy to get those chemical bonds to form. When one bond breaks, it releases energy. Now listen, suppose you had to have a DNA molecule with 1,000 nucleotides. That's not a very big molecule, not a very big DNA molecule. You would have to form 3,000 chemical bonds. Not one of which wants to form, not one of which would form spontaneously before one bond broke anywhere in the molecule. Because if a bond breaks anywhere in the molecule, now you have fragments and you're dead, you see. What would be the probability you'd form 3,000 chemical bonds, not one of which will form spontaneously and naturally, not one of, you see you have to pump a lot of energy into it, before just one bond broke. That would never happen. That would never happen. It's ridiculous. What sort of nonsense are these people talking about, you see? Totally unrealistic. Well, that's not really the big problem. It's a fatal problem. But you see, the real problem is the sequence of these things. The genetic language is determined by the specific arrangement of these purines and pyrimidines. You must arrange them in precise order. Here I have a message in the English language. Now, what did we have to do first of all? We had to invent language. Can the paper and ink do that? Can the typewriter do that? No. The only thing on this earth that can invent language and use languages are human beings. Now, bear that in mind. We say there's information here. Why? Because there's human beings. There'd be, if tomorrow we'd wipe out every human being on this earth, there'd be absolutely no information on this paper. Absolutely. You could not say there's any information here. There'd be no information because there'd be no human beings to read it. Talk about a code in a DNA molecule. Talk about information in a DNA molecule. Why is there information in the DNA molecule? Why is there a code there? Because there's a living cell to read it and to utilize it. You could not have the DNA molecule before life. So what? What if a DNA molecule did just happen to evolve on the earth? So what? The primitive earth couldn't care less. Who, if it had any information in it, if it did code for a protein, who would decode the code? Who would translate that code into messenger RNA? Who would then transcribe the messenger RNA into a protein molecule? You don't have life. You don't have a living cell, you see. There couldn't be any DNA molecule, RNA molecule. Couldn't be anything like that until there were living cells. Just like there could be no such message like this unless there were human beings. But after we do have language, how do we get this message here? Somebody had to arrange those letters in precise order, didn't they? Now, you know monkeys can't do that. If you gave a monkey a typewriter and paper and let him type away, he just types nonsense. They're just dumb things. In chemistry and physics, they're just dumb things too. But in a DNA molecule, there is a code. There is information. How did it get there, you see? That's the big question. Or if we try to imagine we're going to start with proteins, well, we have the same problem. You see, you have these chemical bonds linking one amino acid to the other. You got to pump a lot of energy into those things. I made those type of compounds, both the protein compounds and the nucleotides. I did it because I had a specially devised chemical reagent that was synthesized so that it could donate chemical energy. I didn't just spray it with ultraviolet light or lightning or something like that. Now you have to pump energy into these things. Then, of course, you have to have specific sequence. You see, they have to be arranged in a specific order. Next slide, please. Now here's a protein. This is ribonuclease. Enzymes are chemical reactions. Enzymes are catalysts that make chemical reactions go very rapidly, which ordinarily go slowly. Life without enzymes is an absolute impossibility. These chemical reactions do take place, but you'd have to wait hours and days and weeks and months. And we can't do that. We have to, that food you ate tonight, we need the energy now. And so you have these digestive enzymes in your digestive tract, these enzymes that catalyze this extreme rapid hydrolysis or breakdown of these substances, the food that you got in your dinner. There are all kinds of enzymes. To get life started, you would have to have hundreds of different kinds of enzymes. The most primitive living cell imaginable had to have hundreds of different kinds of enzymes. Our bodies probably have tens of thousands of different kinds of enzymes. But the most primitive living cell would have to have hundreds of different kinds of enzymes. Now, enzymes are proteins. As I mentioned earlier, proteins are made up of amino acids. The building blocks of proteins are these amino acids. Now, there's a real big problem right here immediately for evolutionists. Another one of those insuperable difficulties. You see, if you go in the lab and you generate amino acids, or if they were generated on a hypothetical primordial Earth, they are produced in two forms. All amino acids exist in two different forms except glycine, which is the simplest amino acid. All other amino acids exist in the left-handed form and a right-handed form. Now, you know, if you put your left hand up against a mirror, you see the mirror image of your left hand. Your right hand is a mirror image of your left hand. But you know you cannot superimpose the right hand over the left hand. When you try to do that, you find the thumb of your right hand sticking out in the same place where the little finger of your left hand is, you see. They are different. The arrangement of space is different. That's the way with these amino acids. They exist in these two forms. Now, just for the layman, let's call it a left-handed form and a right-handed form. Their chemical properties are the same. The only difference is if you dissolve them in water, one rotates light to the left, the other rotates light to the right. They have the same melting point, other physical properties, and they are produced in equal quantities, 50% of one, 50% of the other. And if you combine these amino acids to form a protein, the left-handed form combines just as easily as the right-handed form. They combine equally well. So if these things were generated on the hypothetical primordial Earth, you would have 50% left-handed, 50% right-handed, and they combine equally well. Now, in this protein, we have lysine number one. We wouldn't know whether it was left-handed lysine or right-handed lysine. Glutamic acid is number two. It might be left-handed or right-handed. This might be left-handed or right-handed, so it could be left-handed, 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 or right-handed, 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 or left-handed, right-handed, right-handed, you know, any combination like that. They'd all be mixed up. But in proteins today, wouldn't make any difference if you're talking about a bacterium or your human brain. All proteins are 100% left-handed. That is, the amino acids are 100% left-handed. Chemistry cannot do that. The origin of life chemistry absolutely could not do that. There is no way it can choose one over the other. As I said, the lysine might be left-handed or right-handed, and glutamic could be right-handed or left-handed. There's no way to discriminate. The fact that proteins today are 100% left-handed had to be due to deliberate choice, intelligent choice. When I synthesized the protein, it had all left-handed amino acids in it because I reached up on the shelf and pulled down, each case, a left-handed amino acid, you see. If you substitute one of those amino acids with a right-handed amino acid, the compound will have absolutely no activity, none whatsoever, totally inactive. They all must be left-handed. You know, evolution's been working on this problem ever since they started to think about the evolution of life, and they're absolutely no closer to a solution today than when they started. Why? There is no solution. Not from a mechanistic, naturalistic point of view. It had to be by deliberate choice. But let's make the job easy for the evolutionists. Let's assume that all the amino acids on the primitive Earth were left-handed. Let's assume there are only the 20 amino acids that we find in proteins today. The proteins we have today have these 20 different kinds of amino acids. Theoretically, if you could generate amino acids on the primitive Earth, there could be maybe several hundred. Let's make it easy for the evolutionists. Say they were only the 20 that we find in proteins today, and they were all left-handed. Now, we have this enzyme, ribonuclease, 124 amino acids. How, you see, if you arrange those amino acids in precisely that order, you have ribonuclease. This is an enzyme. It catalyzes the extreme rapid digestion of the ribonucleic acid that you ate in your supper tonight. There's hundreds of digestive enzymes that do specific things. This breaks down ribonucleic acid. Why does it do that? Because it has this specific arrangement. Every single molecule of ribonuclease produced by your body has lysine number 1, then glutamic acid, threonine, alanine, lysine, phenylalanine, glutamic acid, arginine, glutamine, histamine, methionine, and so forth. 124 amino acids arranged in that precise order. Now, if we take the same 20 different kinds of amino acids, 188 of them, and arrange them in this order, in which now phenylalanine is number 1 and proline number 2 and so forth and so on, we have human growth hormone. This is not an enzyme. It doesn't catalyze any chemical reaction. It's a hormone. Determines, among other things, how tall we are, or short, or so forth. Now, if you take the same 20 amino acids, 287 of them, in 2 separate chains, the alpha and beta chain, you have hemoglobin, the red blood protein, which binds oxygen. Why does hemoglobin have that specific biological function? Helping to bind oxygen. Why is this thing we have here on the screen human growth hormone? Why does it do that? Why is ribonuclease a digestive enzyme? And each one has incredible activity and abilities because of the specific arrangement of these amino acids. Well, how did that happen? How did those amino acids just happen to get in that order? Well, the evolution says, by chance, just accidentally. It just happened that way. Now you see, it had to be totally by chance, because this idea of natural selection is based upon reproduction. What is natural selection? The fact, supposedly, that some organisms reproduce in larger numbers than others because the good mutant supposedly has gained an advantage of some kind. Now, because it has an advantage in its competition for survival, it reproduces in larger numbers than the original, and so eventually it replaces it. That's what they call natural selection. You could not have natural selection before life because there's no reproduction before life. So it's total chance. Well, one can calculate the probability of something like that happening. If we assume there were only 20 amino acids and they were all left-handed, what would be the probability that phenylalanine would be number 1? Obviously, 1 chance out of 20. Well, that's not too bad, but now we've got to have phenylalanine followed by proline. What's the probability of that happening? 1 over 20 times 1 over 20, that's 1 out of 400. What about the probability we'll have phenylalanine followed by proline followed by threonine? 1 over 20 times itself 3 times. That is 1 out of 8,000. What about the first 4? That's 1 out of 160,000. What about the first 5? 1 out of 3,200,000. What about 188? That's 1 over 20 times itself, 188 times. Let me tell you, ladies and gentlemen, the product is not astronomical. It's super, super, super astronomical. There's no such number in the entire universe. It's more than all the particles in the entire universe combined. Trillion times trillion times trillion times trillion times trillion times all the number of particles in the universe. It's not going to happen. Not on this earth in 5 billion years or 500 billion years. Let me illustrate it something more simply. Let's ask 17 people to line up here in a certain order. Then rearrange yourselves in a second way. And then rearrange yourself in a third way, in a fourth way, in a fifth way. How many times could 17 people line up without lining up twice in the same order? Just 17 people. Well, I know you're not going to believe me, but it's more than 355 trillion different ways. More than 355 trillion. See, I told you you weren't going to believe me. All right. If you don't believe me then, you just get 16 of your friends together and you try it. Well, the universe will be gone before you make all those tries. Just put it on your computer. It's 17 times 16 times 15 times 14 times 13 and so forth and so on. David Coppich's father, Dr. Coppich, done a marvelous job in one of the books that he published on the probability of evolution. Fantastic calculations Dr. Coppich has in that book. Incredible. I mean, when you just get right down to brass tacks, you know, forget your faith and what you think must have happened and start calculating what could have happened, you'll find it's just not possible. Now, let us suppose that one of these molecules, you know, what if it did happen? What if, let's see, by some miracle, it did happen. What would you have? One single molecule of one single protein. That's all. But you've got to have billions of tons each of hundreds of different kinds of protein molecules. You've got to have billions of tons each of hundreds of RNA and DNA molecules and all of that. It's not going to happen. No natural mechanistic evolutionary process could ever accomplish that. And look, I'll give you all the protein that you can think of in any amount. I'll give you all the DNA and RNA molecules that you could think of in any amount that you want, and you'll still not get a living cell. You see, life is more than just a bag of chemicals. A living cell has a membrane, and membranes are very, very complicated things. You've got to have energy factories of some kind and protein factories. You've got to have the genetic material, and it has to be put together in a precise fashion. Just like the mechanism of your watch. You don't get a watch by throwing all the parts in a bag and shaking it up. You never get a watch that way. Each part must be put in precise place. Now, the second law of thermodynamics tells us that there is a universal natural tendency for all systems to deteriorate and run down and decay, to become less organized, less complex. And that's true of everything, isn't it? True of our bodies, true of our houses, true of our machines. It's true of the entire universe. Every star in this universe is wearing out, burning up its fuel. You see, if there is no God, or if God would not intervene, the universe is certain to die, because someday every star in the entire universe would have burned up its fuel, and then the lights are going to go out. There'll be no activity, no life anywhere in this universe. Well, we know God controls, and God is going to intervene. 2 Peter chapter 3, we know that. But naturalistically, mechanistically, the universe is certain to die, because the whole universe is running down, precisely as stated in Psalm 102, verse 26, where it says, the heavens and the earth, just like a suit of clothes, is wearing out. Put in the Bible 3,000 years ago and only discovered by modern scientists during the last 100 years. Now, what I'm saying is this. If you had all these molecules just floating around in the water, they're not going to say to one another, hey, fellas, look, we're just not accomplishing anything. We're just floating around here aimlessly. We've got to get organized. Now, come on, let's get together here. We've got to form some complex system with some meaningful, use and so forth. And let's get organized. No, that's not going to happen. You see, if you had something like that, it breaks apart and disperses naturally. How, then, would all those molecules get together and form something like this? What an incredible faith the evolutionist has. Here's a newspaper article that appeared in the Daily Express of London, August 14th, 1981. Two skeptical scientists put their heads together and reached an amazing conclusion. There must be a God. Wasn't such an amazing conclusion to me, but it was to them, because when they began these studies, they were both atheists. One of these scientists was Sir Fred Hoyle, the famous British astronomer. Now, Sir Fred Hoyle, if he is not the world's foremost astronomer, he's one of the world's foremost astronomers, and his friend, Dr. Chandra Wickramasinghe. Dr. Ramasingh is professor and chairman of the Department of Applied Mathematics and Astronomy, University College in Cardiff, Wales. Also a very well-known British astronomer. Now, some years ago, these two scientists became interested in the problem of the origin of life. The first thing they did was to make some assumptions about the minimum requirement to get life started. Then they calculated the probability of that happening on this Earth in 5 billion years. The probability, 1 chance out of 10 to the 40,000th power. That's 1 chance out of the number 1 followed by 40,000 zeros. Well, of course, that's absolute zero. But they said, after all, the universe has 100 billion galaxies. There's 100 billion stars in every galaxy. Let us assume that every star in the universe has a planet like the Earth. Let us assume the universe is 20 billion years old. Now let us calculate the probability that life has evolved somewhere in the universe in that 20 billion years. The answer came back, forget about it. The probability was so low, it was essentially nil. Sir Fred said the probability of evolution is equal to the probability that a tornado sweeping through a junkyard would assemble a Boeing 747. Well, I tell people, and I tell professors this, I say, if you want to believe that, that's your business. But don't call it science and don't teach it to your students. Now, these two scientists are not Christians and they don't believe the Bible, but they are creationists. They believe that wherever life exists in this universe, it had to be created. Therefore, there must be a God. Now, you see, they made this very logical conclusion. We have established, they said, that a naturalistic, mechanistic, evolutionary origin of life is impossible. There is only one other possibility. It had to be created. Therefore, there must be a God. Did they become creationists because of their religion? Not at all. You see, they became creationists in spite of their religion, because when they began these studies, they were both atheists. Now, here is two scientists, hard-headed scientists, with absolutely no inclination to believe in creation, with every fiber in their bodies wanting to believe in evolution. And they said life had to be created. And you know, these are not the only evolution scientists or converted scientists or people who are, you know, are either not creationists or not evolutionists or couldn't care less, who are saying things like this. Hubert Yockey, this man, as far as I know, is not a creationist. He wouldn't say, I believe in creation. But here's a statement that appeared in the Journal of Theoretical Biology, volume 67, 1977, on page 377. Now, Yockey was worked on information theory. And these people working in information theory are using some of the very same equations that are used in thermodynamics. But what Dr. Yockey did, he said, now we're going to assume that a gene had to evolve by chance and so forth. What would be the probability of that happening on this Earth, you know, under these assumed evolutionary conditions? And we noticed that he said that, quote, the longest genome, that is the longest gene, which could be expected with 95% confidence in 10 to the 9th year, that's 1 billion years, corresponds to only 49 amino acid residues. This is much too short to code a living system. So evolution to higher forms could not get started. Geological evidence for the warm little pond is missing. It is concluded that belief in currently accepted scenarios of spontaneous biogenesis is based on faith, contrary to conventional wisdom. End of quote. What is conventional wisdom? What you read in your textbooks. Life just arose spontaneously, just naturally. That's conventional wisdom. He said, contrary to that, he said it's based on faith, not science. The best you could get in 1 billion years is only one gene, just long enough to code for a protein of 49 amino acids. I don't know of any proteins are that short, have only 49 amino acids. In any case, you could only produce one protein. I would do nothing. And that's the best you could get in 1 billion years. again, based upon hard-headed science. And it shows, you see, that life could never have evolved on this planet. Now, I've debated people on this subject, and I just congratulate them. I congratulate them on their tremendous faith. It's an incredible faith that they have to believe that, in spite of all this tremendous contrary evidence, you see. Now, it's all right to have faith, but it should be a rational faith, reasonable faith. As the Apostle Peter said, that the disciples, you see, had not followed cunningly devised fables when they made known unto us the coming of our Lord and Savior Jesus Christ, but they were eyewitnesses of his majesty. And here today, as modern chemists, we can be eyewitnesses to the creative genius of God. As Rick Womasthing said, we know what an enzyme is and we know what it does, but we don't know why it does what it does. So whoever designed an enzyme had to be vastly more intelligent than you and I. So we should give God the honor and the glory. Honor him as master and creator. The best, most up-to-date statement that we can make scientifically concerning our origin and the origin of life is still, in the beginning, God created the heavens and the earth. Well, thank you very much, ladies and gentlemen. Plan to have some questions,
Yeah.